So I may as well start with some introductions. Like, welcome to the second Cassyni webinar that GigaScience Press has hosted, our first for a GigaScience paper. This paper is hot off the press. It was published this morning by two authors, well, by a number of authors, and we have two of them here who are at Ben-Gurion University of the Negev. Um, I'm very happy to introduce Professor Esti Yeger-Lotem and Dr. Michael Fire, who work at different departments there. Esti is head of the Department of Clinical Biochemistry and Pharmacology at BGU, and Michael works at the Software and Information Systems Engineering Department. And also runs the Fire AI lab. So this is a really interesting study from a kind of biological and scientific perspective, but also there's an interesting story here about how algorithms taken from wider AI fields can be bring very interesting biological insight. And so this webinar is a great opportunity to not just discuss the paper, but kind of pick apart these things that may not be quite so obvious from the actual paper. So I think Esti is starting first. So
Yeah,
please take it away, Esti.
yeah. So I hope you can see it. Hello, everyone. I hope you can see my screen. I'll just do something here to hide meeting controls. Okay. So hello, everyone. I have a computational biology lab at BGU, as Scott just said. And what I'm going to do is give you like a short introduction to what we've been studying in the recent years and what we've been doing. So one of the things that fascinated me is how we have, from looking at the human genome, we have a copy of the DNA. This DNA is present in almost all cells throughout the body, and this copy is the same. However, when we look at tissues and cell types and phenotypes, they tend to differ a lot. And the question is, how does it work? How do we get from a single sequence to a diversity of tissues and cell types and repertoires? And this question becomes even more intriguing if we look at how genes are expressed across tissues and across cell types. And this is what I'm showing you here based on data from GTEx. So actually, most of the genes are not tissue specific. And now we know that they're also not cell type specific. In fact, they tend to be expressed across many different tissues and cell types. And therefore, it makes it really hard to understand how we get from one DNA to this diversity of tissues and cell types and phenotypes in general. So with this understanding that this phenomena is not due to the expression of single genes, it's actually more like a network phenomena, we developed throughout the years all kinds of tools and views in order to understand this. So we've been working with protein interaction networks, defining what we mean by tissue-specific networks. We've been looking at genetic modifiers and how they are expressed across cells and tissues. We've been looking at gene sets and cellular processes and even fundamental cellular processes that are supposed to be present in every cell throughout the body. And yet we analyze them and they do have some differences. And we developed all kinds of tools, and these are available online, and you can go to my lab's website, which is here. Now, one of the enigmas that we've been studying a lot in the recent decade is the tissue selectivity of hereditary diseases. So what is a hereditary disease? This is a disease that is caused by mutations that are in the germline, meaning the mutations are actually present in all cells throughout the body. Now, we analyzed many of these diseases. This is together with my student, then Ruth Barshir. And we found out that many of these hereditary diseases tend to manifest clinically in specific tissues and actually not throughout the body. And this is kind of surprising because, as I said, the mutation is in the germline, so it's present throughout the body. However, the disease manifests in specific tissues. And as an example for this is a familial mutations in a gene called alpha-synuclein, which lead to Parkinson's disease, manifesting in a very, very specific region of the brain. So not in other parts of the body, they are not affected. Other parts of the brain are also not affected, at least until the disease progresses. So the question becomes, so how does it work? How does a mutation that is present throughout the genome or throughout the different cells and tissues, etc., how does it manifest clinically in a very specific place in the body? Now, as I said before, this is not because the causal genes, the germline or the disease genes, it's not because they are expressed in specific tissues. Actually, many of them tend to be expressed across all tissues. And therefore, we started looking at this phenomena and we started analyzing computationally. And you kind of flip to the question. So instead of asking, why does Parkinson disease occur in this specific region of the brain, we now ask a different question, and we ask, why does this disease does not manifest in many other tissues and cell types that also express the aberrant gene, the disease-causing gene? And to understand why this view of why something happens and why does it not happen, why is this view powerful? We could look at these images and kind of play finding the differences. So if we would just look, for example, in this alpha-synuclein inner brain region and look at what's around this gene, what does it interact with, etc., we would see many, many details. However, if we look at what happens in other tissues and other cell types and other regions of the brain, many of the details would be the same. So if they are the same and these regions are not affected, it means that these details are not important for the disease. And now we can try to find the differences and find the things that actually distinguish the affected tissue from the unaffected tissue. And this leads us to a much smaller search space of the differences that manifest only here in the disease-related tissue and not in others. And this can lead us perhaps better into understanding disease mechanisms. So if you're interested in knowing more about this, we have this review published in Nature Reviews Genetics now 5 years ago. Now, with this enigma in mind, a few years back, we applied differential networks analysis to tissue protein interaction networks. So what do I mean? Basically, we wanted to look at protein interactions because this is how protein actually do their parts or do their role. And we wanted to study protein interactions across different tissues. So looking at a specific tissue, we looked at all the protein interactions that were present there. For example, let's assume these are the protein interactions. And then we decided that we are going to add a weight to each of the edges here. And the weight would be a combination of how much this gene is expressed and how much this gene is expressed. So if they are highly expressed, the weight would be high. If one of them is lowly expressed, the weight would be lower. And now we wanted to make this view also differential. And therefore, we computed what's called the median network. Here, every weight is actually the median over the weights across the different tissues. And now we subtracted this median network out of the network that represents what happens in specific tissue. And thus leads us to the differential network of that specific tissue, of tissue I. So now, if this edge here has a positive weight, it means, or we can infer from that this interaction is much more likely in this tissue relative to the other tissue. Or else it could be much less likely in this tissue relative to other tissues. So we published that. This is what's called the differential net database of human tissues. And more recently, we also applied this view of differential network analysis to better highlight tissue selective processes and genes related to human diseases. So this was a short introduction to what our current paper is about. And I just say that this was done with the help of two amazing then they started as PhD students, but now they are doctors. So Dr. Dima Kagan and Dr. Juman Jubran. And now I'm handing this to Mickey.
Great. Thank you. Wait a second. Can you see my screen?
Yes.
Great. So my name is Michael. Thank you so much, Scott, for presenting me. And I'm from a different part of the campus from Esti, the opposite side. I'm more working with social networks, data, AI, more in engineering, applied AI, for example, solving different problems. During my PhD, I did many studies about how to detect fake profiles in online social networks. So the idea is the Esti group, they are doing something with bioinformatics. I don't know almost anything about bioinformatics, but we both joined that we analyze networks. So in my first work in my PhD, one of my first works and studies, we analyze fake profiles, and fake profiles are the bad actors in online social networks. If somebody tried to steal your money, try to send you viruses, spam, probably using fake profiles to do it. So there are different approaches to use to detect fake profiles, and we presented several of them. The idea is like pretty simple. You have a network, you have regular users, you have malicious users, fake profiles usually, and you try to learn what is the difference between fake profiles and regular users. Use supervised learning, for example, different methods, and this idea of using supervised learning to differentiate between regular users and fake users is pretty simple. And usually it works, but the problem is that it's like a cat and mouse game, and over time you need to present more and more solution and more advanced solution to detect fake profiles, because this is like I think multi-million-dollar market to create more, fake profiles. So they all the time they become better and better in hiding themselves. So over time we analyze different ideas to how to detect fake profiles. For example, if you look at fake profiles, most of them are choosing the victim in randomly, not knowing. Most of them, many of them using, trying to detect the victim in random, and they are connecting to many communities, many groups. For example, in LinkedIn, they can connect to many different groups that are not related, while regular users are usually connected to small number of groups. And the idea here is that if you look at the network topology of fake user and regular user, they're different. And this is another solution we developed to detect the communities. And a couple of years ago, we developed a new type of solution that tried to detect fake profiles and differentiate them from regular users using the structure of the network and machine learning using the links. The idea is that if you are in social networks, many cases, for example, in Facebook or LinkedIn, the network like just suggest who can be your friend, and give you high likelihood for people that are going to be your friend and people that are not supposed to be your friend. So if we can measure if somebody is your friend and somebody is not your friend, we can find fake profiles, because fake profiles, if we look here, fake profiles have many improbable links. The problem is how I can find improbable link, or links in this case, and there is like a classical problem in social network, in everywhere, that called link prediction. The idea in link prediction is that you create algorithms to predict if link between two users exists or doesn't exist. So you can use different methods. There are thousands of papers about link prediction, and you can use this algorithm to take a link and predict if the probability of link to exist or not to exist. And what we discovered that regular users have only few links that with low probability, and fake users have many links with low probability. And from this, we move to biology. This is the same idea. We can use the same idea to find interesting genes, or in this case, proteins. So the method we use with Esti is pretty much the same thing that we use in online social network. Only I think this method is even better, because we improved the technology of deep learning improved, but the same idea is it's pretty much the same. The idea is that interesting proteins, usually I use users, but in this case we talk about proteins, behave differently. So pretty much we start from the beginning. We have like several steps to how to find these interesting proteins. The first step is collecting data. Similar to Esti, we use their data set with 17 human tissues, and the idea is that we have PPI networks, protein-to-protein interaction networks, with the same number of proteins and interaction. The difference between this and the online social network, in online social network, we only add if a link exists or doesn't exist. In this case, we have a weight for each interaction between proteins, between -1 and 1. That is the likelihood of a two-gene expression. I don't know, Esti, you need to help me with this, but it's pretty much the likelihood of the interaction between two proteins related to this. And the idea is the same idea. We want to know if the interaction supposed to be there or doesn't supposed to be, and to try to measure it. So our first step is collecting data. Our second step is training a classifier. So in the fake profiles, we trained a classifier that can predict the probability of a link. In this study, we use a classifier, in this case, weight estimator. This is not a classification problem, it's a regression problem. The idea is that we can create a weight estimator that can predict the weight of interaction between -1 and 1. So it's basically the same idea. You take a lot of examples, train a weight estimators. We have new type of algorithms that were developed in recent years that can embed nodes and interaction, in this case links. And basically you take interaction between two proteins and can generate a feature vector that represent this interaction. The feature vector have weight, the actual weight that was measured. And using this and supervised learning algorithms, you can create something like really nice that can predict the weight of interaction. This is like very basic idea how to predict weight of a links using deep learning. And it's not only for this study, there's pretty much many studies that doing something similar. But in our case, it's actually worked, and we tested different type of settings to predict the interaction. Next, using the weight estimator, the next step is predict the actual weight. So if in the next step we have the actual weight, we can use the weight estimator to predict the weights using our algorithm. So for each network, we have the actual weight and predicted weight. The actual weight is in orange, the predicted weight in purple, and you can take both of them and use it to predict the error, not the predict, to measure the error for each interaction. So what we actually predicted and what the actual error, and we can use it. And then we get this network. This network is the same network as the beginning, only with errors instead of weights. And using this network, we can use and measure different feature for each protein, or each, in this case, each node, and measure different feature about it. For example, the maximum error, the mean average of error for each vertex, for each node or each protein, standard deviation of errors, and so on. So basically, we took the network for each tissue, created new network, and this new network for each protein we have different feature that is based on the errors of our prediction. And what we discovered, this is really interesting, because using these new features, we can rank each protein according to how abnormal each protein is. For example, we can use the maximal error for rank the protein according to the maximal errors. We can use different algorithms that get feature and predict anomaly score, and so on. And there are different methods. And what we discover that using this method, our own method, WGAND, that we developed, is really good in detecting different type of interesting proteins in Mendelian disease or other diseases, better than other methods. And what is really cool about our method is that it's generic algorithm. We use it like for predicting interesting proteins, but in the same way we can predict fake profiles or predict abnormal nodes or vertices in transportation network, in medical network. It's like really generic and can use for different methods in different domains. What also interesting is that our method is also reproducible. We publish in GigaScience, and we'll have to publish there. But one of the conditions that everything is like open, and our code is open, our dataset is open, everything is open, and you can use it, and it's supposed to be really easy to use. If you use them, please send us an email to say it worked or didn't work, and you found something interesting. And that's it. So basically, taking from the world of fake profiles to the world of proteins that I don't have a clue about it, similar ideas like can work and in different domains. It's really interesting, and all this research was really interesting way to explore new domains with the same ideas that from fake profiles. And that's it.
Thank you both. That's a really fascinating inside approach. So we have a little bit of time now for questions. People can post them in the chat. The nice thing about Cassyni is you can post them for the next 24 hours as well. So if anybody asks questions in the next 24 hours, we can answer them as well, hopefully. I would like to ask one first. Oh well, yeah, so we have a question from Diana. And this was kind of the question that I wanted to ask as well, actually. Diana is asking, I want to know how you two met to work together. Because you're working you come from two different fields, you come from two different parts of the campus. How did you both meet and start collaborating?
Esti, you and me.
You can go.
So basically, there was a mutual friend in my department is also working with biology, and there was a grant proposal, like grant call, small grant call, for multidisciplinary research, and she connected us both. And again, we won the grant, so we started working, and then we won another grant, I think, right, Esti? We have two grants, yeah.
Yeah.
So
Yes.
in BGU, we have many small calls for multidisciplinary grants and try to connect people from different departments.
And actually, we look at similar things because we've been looking at protein interaction networks, and Mickey was looking at networks. So the combination of the two, and his idea of fake profiles, and our idea that certain proteins have specific roles in specific contexts and not in other contexts. So these two ideas combined. But also it took us a long time to finalize this study. It's not as easy as it seems.
After you see the road how to get to one point to another, it's really easy. But the way was really long. By the way, when we submitted the grant, I never imagined that it's going to work. All right, let's try to win this, and maybe it's going to work, maybe.
And there's another question from Diana. Have people done this research before? Have people used this kind of approach before?
To my best of my knowledge, no. We published a paper about fake profiles, and that's it.
And that was a decade ago, right? This was work algorithms you were working on. The example you put was 2014, right? Right.
Yeah, this is a decade ago, but we have recent one, Dima also, and the recent one is generic. I think it's a couple of years ago. So that's like the end of his master degree. Yeah, it's a decade ago.
Yeah.
It was a master thesis. He already finished his PhD a year ago.
But the history of biology, so many of the algorithms and things that people use, even like going back to the very beginning of molecular biology, it was physicists who kind of switched to biology, algorithms used for engineering and physics and other applications. It took a long time for then people saying, oh yes, we can use this for genome assembly, we can use this for alignment and things like that. But there's been big delays. But do you think machine learning, just the explosion of, AI, do you think that process is speeding up? Do you think there are things are coming from multidisciplinary areas much faster now?
They definitely do. For example, we've been using machine learning in order to identify variants that are causal for diseases. And that's something that we could not have done before. Doing this, like taking AI or machine learning and biology and all the data sets that we have, it's not an easy task, but it becomes much more feasible. So you can think of something and then get it done. I think both at a systems level, meaning across many components, across many tissues and cell types in our case, and also much faster.
And do you see any skepticism from the more pure traditional biologists about because this is a big leap from social media to to protein, protein biology and the like. Have you, from your colleagues and from feedback from presenting this, have you seen any pushback, or are people accepting just with machine learning and AI approaches getting just more ubiquitous? Are people more ready for this kind of um, type of
Yes.
research?
So it really depends who the audience is. So if these are people that are like perhaps more used to machine learning, etc., and then they are much more acceptable. And we always show these, as Mike just showed, the statistics that shows that we are more successful than other methods, etc. But also in biology, people would, in the end, wait for the wet lab experiment. And so that would be like the total proof, even if sometimes it's more like cherry picking, but still, this is the wet lab proof is always something that is best to have, but it's really hard to obtain.
So we have a question from Bushra, who's saying, firstly, I want to say this research is really good, especially the idea of detecting the concentrated area of genetic diseases using the algorithms used for fake profile detection. My question is, how do we verify the results that we are getting from the study? Were the results lab tested? So yeah, this is a validation question.
So we did a few tests. In one of them, we looked at what we call the anomalous nodes in each network, and we asked across these top 10, let's say top 10 anomalous nodes, how many of them are, for example, related to disease that manifests in this specific tissue, and how is this compared to random, how is this compared to other algorithms, and we see that we have much better chances of picking these disease-related proteins. We did a similar analysis, slightly different, but also with proteins related to tissue-specific processes. So again, this was done by manually curating some of them and then seeing or checking whether they tend to occur across the most anomalous nodes in the network. And again, they do, and they did so both better than random and better than another method. And then we also use some kind of a computational assay, which is different, which is like how well are they predicted to be related to tissue-specific processes or diseases? And again, we got good results. So can we pick everything? No, no, it's not a perfect method, but it gives much higher chances than current methods in identifying these proteins.
And so yeah, there's the more downstream validation part, and so the obvious question is, what's next? You said you've both got another grant. I don't know if you can say anything about, or if you're able to talk about what your plans for the future are.
Each of us waits for the other to answer.
Yeah, yeah. So I start. We got like this was the grant for this. We got like small grant, and then we got a bigger grant for this research because it took long time. I think right now maybe looking at LLMs. I don't know. It's like I don't know if with this research, but LLMs like in everywhere, like big language models are everywhere right now. All my lab like took 10. Every research is right now using LLMs pretty much, or something with deep learning, but LLMs and deep learning. So probably this is the new direction.
Yeah. Also, in terms of biology, we could always use more data, and the method that seems to be like the top now is single cell technologies. So again, the idea is to apply the same kind of idea into, but using single cell technologies as part of our data sets.
What about spatial data? What about like GigaScience? We're publishing a series at the moment of spatial omics data, and we see that there's a huge growth
Yeah.
in that tech. Is very, very data rich. So do you think that this similar approach could work with that type of data?
Yeah, I totally agree. That's why I said single cell technologies in order
Yeah.
to include spatial data in, because it can include expression data, spatial data, just as you said. So yeah, I think biology wise, this is like the new front, or like the most exciting front now.
No, it's exciting times, definitely. So I don't know if there are any final questions. We've kind of hit half an hour, the half an hour scheduled. I don't know if you have anything you'd finally like to say. Um, if if not, then thank you both for presenting your work for a fantastic paper that you gave to us at GigaScience. We got a message from Diana and some of the attendees saying thank you. So yeah, thank you so much. This video will hopefully be archived and available for others to view. And yeah, keep up the good work, and we're excited to see where this goes in the future. Thank you so much.
Yeah,
Thank you for hosting us.
yeah, thank you for everything.